reorganization, so that they tended to be about 2 to 5 angstroms RMS from the deposit. And so we refine these with this low-resolution refinement protocol in Rosetta and compare the results versus Phoenix Refine. And I'm just plotting. So for each of these 26 cases, it's just a single point in the scatter plot. The x-axis is the result on Phoenix in R-free and RMS, and the y-axis is Rosetta. So you can see there's a lot of cases kind of on both plots along this diagonal. So these are cases where down here both methods perform equally well. Kind of up here, both methods are unable to capture this domain motion. But there's also quite a few cases down in this region where by using Rosetta's force field and this combinatorial sidechain repacking, we're able to solve the structure where the standard refinement couldn't. And I'm going to just highlight one of these where Rosetta performs particularly well. This is this 3 FPS is the target, and we're using a template 2 ZVG. And so what you can see is that over most of the structure, it actually superimposes pretty well. In fact, this structure is identifiable by molecular replacement, but there's one domain that's largely reorganized. And if you look at the density, it's not totally clear how you should refine the structure. But after this sort of iterative real space and reciprocal space refinement with these combinatorial sidechain optimization that's done by Rosetta, it snaps right into place. R&R-free is in line with the deposited structure, although there is some kind of local changes, some loops that are in different conformations than are in the deposited structure. And finally, we also tried this on a blind case. So crystallographer had a 3 angstrom data set that the kind of refinement got stuck. And so we wanted to see if we could use MR Rosetta to solve that as well. And indeed, we were able to take, so there was a closed homolog. After rigid body refinement, it had an R-free of about 42%. After Rosetta refinement, at about 28% R-free, and kind of maintained good model geometry. Okay, and so then the second part of my talk, I'm actually going to talk about cases where the initial molecular replacement search fails. And so here, I'm going to talk about ways that we've improved homology modeling in Rosetta so that we can actually that may be able to help us better find, better place models than using the template structures alone. And the basic idea is that the basic new things in Rosetta are that we've managed to incorporate multiple templates when building a single model, as well as sort of improved methods for backbone remodeling. So the way that Rosetta homology modeling works, and in fact this is the way that MR Rosetta also builds models into density, is that basically given the amino acid sequence of our protein, we do a simple sequence alignment, and so generally use HH-pred. For distant cases, alternate alignment methods like Sparks and Raptor are really good at finding very, very distant, or for identifying and aligning to very distant homologs. But HH-pred is generally very, very good. And then, so from these sequence alignments, we identify a bunch of templates. From those templates, we can then thread the target sequence onto the template structure, create these threaded models, and then do this kind of rebuilding and refinement that I talked about earlier, except without density data. This gives us then sort of a number. We build a bunch of models for each of these, and then can then use Rosetta's energy function to kind of pick from among them. And the basic idea is that one of these templates is better than everything else. And so if we can identify that template, then we'll just use those models. But really what happens is that in a lot of cases, no one template is best, but rather certain templates are better in certain regions. And so we've developed a new method, this multi-template modeling, that allows us to sample, kind of in a Monte Carlo trajectory, to sample pieces from different templates and recombine them into a single model. And so as an example of this, I'll show you. So this is one of the templates. And this is work done with Yifan Song in the Baker Lab, I should point out. So one of the templates, one of the structures that we tried to solve is shown here. And so there's 2 really good templates for this structure. One TZA captures this hairpin correctly, but sort of this back sheet is incorrect. The other, this one LMI, misses the hairpin, but this back sheet kind of almost perfectly models the target. And so by not just modeling these fragment insertions, but also by allowing different pieces of the templates to recombine, we're able to capture both of these features in our selected model, both this hairpin and this back strand. Additionally, excuse me. Additionally, this required that we develop sort of new ways of modeling, a new way of rebuilding unaligned regions and of closing gaps in the alignment rather than the fragment insertion and CCD closure that I showed before. Again, I kind of showed this method before. You basically do fragment insertion, CCD closure. The problem is now that we're combining multiple templates together, first of all, the sort of unaligned regions are different depending on what combination of fragments we have. So at any given point in time, we might have different kind of segments to rebuild. And additionally, kind of when you stitch two different templates together, at that junction point, there tends to be a gap that you want to close as smoothly as possible. And so we developed this kind of fragment stitching closure instead. And so the basic idea is that instead of kind of keeping the template fixed and doing the fragment insertion, we just identify a fragment that tends to match things at the endpoints reasonably well, like this fragment shown in red here. We superimpose that in place and then do a global minimization of the whole structure using the low resolution energy function. And so it's actually a smooth variant of this centroid energy function. And so we globally minimize this, and this allows not just the fragment to snap into place, but also these end positions to melt a little bit. And so, again, we developed this mainly for, to deal with some of the challenges posed by this multiple template modeling problem. But it actually turns out that this works better even in the single template case. And so I have just a real quick example here. So we have this template in blue, and the native is in black here. And so this sort of fixed backbone modeling that we did previously, where we just rebuild the gaps and then relax the whole structure with the all-atom energy, kind of doesn't really do a good job of recapitulating this curved sheet here. However, if we do this fragment stitching strategy that I showed, then it actually does do a very good job recapitulating the native. It's not totally clear why this works better, but there's a number of possible reasons. So first of all, it allows, so as we're doing rebuilding, it allows subtle changes to the template structure itself, which I think in this case probably is the key factor. It also, again, when we minimize, so the centroid energy in Rosetta, because we don't have side chains, the centroid energy in Rosetta, it really features terms that optimize backbone geometry, backbone hydrogen bonding, and like backbone sterics. And so it may be that preferentially optimizing these just gives us better models overall. And finally, one problem with the geometric closure is that you kind of have this junction point where the gap comes together, and that's not fragment-based, that's just geometrically determined. And so that's kind of non-protein-like. And so this new method, though, avoids these non-protein-like because we're just minimizing things close. It avoids these non-protein-like junction points. So when we put everything together, the protocol looks something like this. So the native structure is shown in this gray color here, and the template is shown in blue. And so you can see there's this large gap here. But so here we're sampling pieces from different templates and then following that up with these fragment stitching moves and centroid space minimization. And you can see that we're able to, after a couple cycles, kind of close these gaps. Okay, and so then the last part, so we use this approach. So CASP is this biannual this blind structure prediction competition, and it just concluded back in September of this year. And so this multi-template modeling protocol was used for the Rosetta homology modeling this year in CASP. And the protocol seemed to work pretty well, particularly in close homology modeling cases. So what I'm plotting here, so these are all the different servers, the CASP server models that were used. And this is, and so what I'm plotting is for each server on the comparative modeling cases, how often a server had the best model overall. And so here you can see that the R group server did particularly well under this measure. And then also in black, I'm plotting if you just consider the very, very close homology modeling cases. And these are cases where like sequence alignment probably isn't a concern, but the sequence alignment is straightforward. And so this is plotted as a function of GDT, which is looking at sort of the backbone kind of accuracy of the models. And so then what I'm plotting here is I'm comparing just the other servers to the Baker server on these models shown in black. These very close homology modeling cases. But actually, if you look at this in terms of, but this is just a backbone-only measure. And so actually, if you look at this in terms of side chain recovery measures, and I'm just showing two of these. So this is a side chain tip atom GDT, where this computes basically the sort of the C alpha superposition. This takes, instead of the C alpha, a tip atom of each side chain and computes the same measure. You can see there that Rosetta is actually doing significantly better. And again, if you look at the fraction of hydrogen bonds recovered, you see the same thing. And so we're much better at accurately recapitulating side chain conformations. And so then here's, I'll just very quickly walk through this. But here's some of the results. So in black is the native structure. The top row is the templates that were used. Excuse me, these are the best templates that were available for each of these three cases. In green, I'm showing the Rosetta model that was built. And in blue and purple, I'm showing two of the other best servers and the models that they built. And so what you can see, and I'll just talk about this one on the left here. So what happens here is these two helices, to accurately kind of capture their side chain packing, you really need to let this helix move off template somewhat. And so the side chain, or excuse me, the backbone stitching and the centroid minimization that Rosetta does is able to capture exactly that. We get the helices placed almost perfectly, and the side chain packing is almost perfect. This is a little bit off. But you can see that the other methods just can't make that movement from the template to the native structure. And you see that again in these two cases as well. I mean, also in this case on the right, the backbone, to get these two hydrogen bonds, to recapitulate these two hydrogen bonds, really this backbone needs to move. Rosetta is able to capture that and get these two hydrogen bonds. But the other methods kind of, they move things a little bit in the right direction, but they can't completely get these close enough that these hydrogen bonds are recovered. So then, again, because of this, this approach should be particularly well suited to molecular replacement. And so we need to do further testing on this. Now, I've just kind of informally tested this on about a half dozen cases. And these are all cases where I initially tried MR Rosetta and was unable to get a solution. And in fact, of these half dozen or so, two of them we were able to get solutions using this hybridized. And again, these were all cases where none of the templates were able to place the molecule correctly. And the first of these, you can see that again, this is starting to get to very distant sequence identity. So here we're about 17% sequence identity. The templates are all shown here. In black is the model that was able to phase the structure. And this is all fairly small scale sampling. This was just constructed sort of overnight on a 20-core machine. So it's not super significant amount, maybe a CPU week of time. And again, actually in this case, this model was just able to Phaser found a pretty easy solution for this. Automatic chain tracing was then able to take this to basically to completion. This case was a little bit more difficult. This was 2.2 angstrom data. The closest template here was 16% sequence identity, but there was a very large number of templates that were available for this. We were able to build this model in black, we were able to get a very weak molecular replacement solution for this. Then using MR Rosetta, we were able to get this good enough that automatic chain tracing could get this to close enough that the crystallographer could solve it. So in conclusion, I guess, so first of all, combining Rosetta's energy function and Rosetta's sampling methods with low resolution or noisy or poorly phased experimental density data allows for us to kind of better fit models to data while maintaining sort of physically favorable interactions, like maintaining favorable hydrogen bonds and favorable core packing from the template structures. Additionally, the multi-template modeling, it provides models that certainly have better side chain recapitulations, and they may be more well suited for molecular replacement than other approaches. We need to do further testing with this. And also, I guess another thing that we're looking at, I'll just go through this really quickly, but basically, we've shown that this newer multi-template modeling works better in the absence of experimental data, and so there's no reason not to believe that it would also work better with experimental data. And so we, again, tried this multiple template modeling in the context of low-resolution refinement. And here, in this case, so this is one of the cases from the low-resolution refinement set. Kind of the structure's pretty much correct, except for like locally there are these fairly large changes from the template to the target structure that they're just not, through minimization alone and sidechain repacking, they're just not able to go from the structure in salmon to the structure in blue. But by doing this sort of multi-template modeling approach, I guess mainly the fragment stitching approach, we're able to make these fairly large-scale backbone changes. However, this did take, I should mention, this took a fairly significant amount of sampling, about 2,000 models, which was, I think, several CPU days. All right, so finally, the Rosetta code is available from rosettacommons.org. It's free to academic users. In terms of the 3 different things I talked about today, Rosetta MR is already available. It's in the current Rosetta trunk. There's also tutorials available. So if you download the Rosetta demos release, there's 2 demos. There's a Rosetta MR demo, and there's also a Rosetta density demo that describe different things that you can do to do sampling with experimental density data. The crystallographic refinement will be available in the next Rosetta release, which should be hopefully mid-March of this year. The multi-template refinement, there's limited support in the current Rosetta. If you're really, really interested, you can email me and I'll tell you exactly what you need to do to get things set up. Again, in the next release, there's going to be full support for this, plus tutorials, plus some scripts to make things a lot easier to run. Also, it's currently the backend to Rosetta. So Rosetta is the Rosetta structure prediction server. It was really for this cast. Large parts of it were rewritten. And not just changing this backend, but also trying to greatly reduce the turnaround time of structures. And so now, for close homology cases, you should get models in less than a day. So I definitely recommend trying that out. And finally, I want to acknowledge, so from David's group, Yifan and Ray and Mike all have given me a lot of help for various different phases, mainly with the hybridization, or with the multiple template modeling stuff. I also wanted to thank particularly, so Paul Adams, Nat Eccles, Jeff Head, but particularly Nat for the crystallographic refinement, and Tom and Randy for the Rosetta MR work.
Questions for Frank? Randy?
Sorry, I shouldn't sit up all the way here when I want to ask a question. Frank, the multi-template stuff that you're talking about.
Yes.
Could you use that alternatively for a single template but multiple alternative sequence alignments? So if you had suboptimal sequence alignments, because Rosetta gets stuck quite often because the sequence alignment is wrong.
Yes. Yeah, you could, and Yifan has looked at that a little bit. I guess one of the problems is when the sort of sequence alignment is wrong, oftentimes you have to go if you generate alternate sequence alignments, the second one is also often wrong. But yeah, so Yifan has looked at that, and that definitely, that works. I mean, that definitely works.
Yeah, on the stairs there.
Could you add extra energy terms representing data from other techniques like SACS or NMR?
Yes. Yeah, you certainly, I mean, that's actually in Rosetta, I think all those cases would be straightforward. If you had sort of multiple sources of density data, that would be a little bit trickier, but it's doable. Yeah, all that should be workable in Rosetta. I think kind of getting the relative contributions of each might take a little bit of tuning to figure out sort of how much to weigh one versus how much to weigh the other. But you certainly, all those data sources are usable, or certainly a large number of those data sources are usable by Rosetta.
If there are no more questions, could you all be back here for the next session at 22? And I'd like to, oh, sorry, there's one more quick. Gabor.
So I'm Gabor Bunkoci from Cambridge. And so the models that Rosetta is producing are truly amazing. So they are really accurate, at least on the pictures that you are showing. And
yes.
My question is that can you also predict your confidence, or in other terms, the error in this model?
So
in RMSD, what deviation do you expect for a certain atom or residue or whatever?
Yeah, so that's actually, I probably should have shown something like that, but generally you can get a really good sense of that from deviation between multiple models. So for example, the multi-template stuff, generally, for CASP, many models were built, but generally 100 models is sufficient to kind of cover conformational space sufficiently. But if you look at the per residue deviations among those 100 models, that really gives a good sense of how, and I think that's very closely correlated to the actual error. And I certainly could collect that data.
Okay, as I said, could you be back at 22? And I'd like to thank all this afternoon's speakers again.